Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So let us begin with the first lecture. Today I will introduce you to the fluorescence microscopy and its applications. So let us begin with a brief history of microscopy, and before going into the history, let me define what is a microscope. So microscope is an instrument designed to produce magnified image of an object that is too small to be seen with the naked eye. So very first microscope was developed by the British microscopist Robert Hooke, and that looked like this. So here you can see he developed this microscope in 1670, and here you can see the light source is separate from the microscope body. So the light is projected onto the sample, and here you can see a tiny sample holder where you will put your sample, and this is the objective lens, so which will produce the magnified image of the object in the eyepiece. Then several developments took place, and Powell and Leland produced little more sophisticated microscope in 1850. It looked like this, where the light source is integrated, and now you can actually focus the light to the condenser of the microscope, and that can be used for making the magnified image of the object. Then in 1930, the first commercial microscope was launched by the company known as Carl Zeiss, and this is the first commercial microscope. And after that, several more modifications took place in the microscope. And eventually in 1998, you can see a highly sophisticated fluorescence microscope was made commercially available by several manufacturers. So there are four branches of microscopy known as optical microscopy or the light microscopy or the fluorescence microscopy. So the first branch is the main focus of this course. But there are several other types of microscopy also, which are known as electron microscopy, scanning probe microscopy, and X-ray microscopy. So in this course, we are not going to talk about this electron microscopy, scanning probe microscopy, and X-ray microscopy. We will be only talking about the fluorescence microscopy and the light microscopy. So on this slide, I have shown a comparison between a very simple student's microscope or the school microscope, which I am sure most of you must have seen this kind of microscope either in your school or somewhere in the pathology laboratories. Okay, and we are going to compare it with the highly sophisticated fluorescence microscope, which is used for research, and it looked like this. So generally this kind of sophisticated research microscope, they comes with the computer which has the software to operate the microscope, and what are the images are produced, those are being recorded. The student microscope generally cost few thousand Indian rupees, whereas the fluorescence microscope that is generally being used for research ranges from few lakhs to few crores. Okay, and by the end of this course, you will become an expert using this fluorescence microscopy for the most of the research applications. So now let me show you which kind of images can be produced by the student's microscope. Okay, so here you can see the student microscope is available in different configurations, which are known as bright field microscope or the dark field microscope or the phase contrast microscope. Okay, and using all these different configurations, you can produce images like this. So here you can see the bright field image where you can see the cell morphology, you can count the number of cells, and you can just see the size and shape of the cells. This is a dark field microscopy. So here the background is dark and the objects are bright, and again you can just see the size and shape of the cells, you can count the number of cells, and you can just see the external morphology of the cell. This is another bright field image where you can see the distribution of vascular bundles in the plant tissue. This is an example of the phase contrast microscope where you can see the cells. Again, you can see the size and shape of the cell, you can count the cell. It just gives you a little better contrast compared to the bright field microscope. And this is another image of the dark field microscope. As you can see, the background is dark and the objects or the cells are bright, and again it shows you the size and shape of the cell and external morphology, and maybe you can just count the number of cells. So using this kind of traditional microscopy methods, what you can learn about the cell. So you, as I mentioned, you can just see the size and shape, you can count them, and the external morphology you can see or learn about the cells. But what you cannot see in these images, if I ask you, can you see whether the cells are dividing but the chromosomes are segregating or not? If the transcription is active or not? If the translation is going on or not? So those kind of intracellular information you cannot see using this basic school microscope. To visualize those kind of intracellular processes, you need highly advanced research microscope, which can produce fluorescence microscopy images like this. So here I can show you what kind of images can be produced using the research microscope or the fluorescence microscope. So here you can see the cells are dividing, and the chromosomes are stained in blue with a blue color dye, and the microtubules are stained with orange color dye. So using the fluorescence microscope, you can actually visualize the intracellular processes that, okay, the cells are dividing, these cells are at metaphase or anaphase. In this image, you can see three different structures are stained in three different colors. So let's say the nuclei are stained in blue color. The microtubules, which are proteins, they are labeled in green color, and the F actin, which is again a protein, is labeled in red color. So using a fluorescence microscope, you can see the co-localization between the proteins and the cell organelles, or you can visualize the different proteins and cell organelles by different staining techniques that we are going to study in one of the next class. Also, this is the most advanced fluorescence microscopy technique, which is known as single molecule imaging. So where the protein molecules are labeled fluorescently in the live cells, and you can study their dynamic properties, that how fast these proteins are binding to their specific target sites, or how fast they are moving inside the cell nucleus. So basically, the fluorescence microscopy makes the intracellular processes visible. I will show you a very good example. I have provided the link of from the YouTube here. You can actually see the entire zebrafish embryonic development at single cell resolution. Let me click this video. So in this video, you can see the zebrafish embryonic development at single cell resolution. The membrane protein is fluorescently labeled, uh, and also now you can see some yellow colored fluorescent protein is coming up. So those are the histone proteins which are labeled, and you can see how from a single cell this multicellular embryo developed. So this is the beauty of fluorescence microscopy, that you can capture such kind of intracellular processes uh, in live cells or the live tissues. So let us go back to the slides, and now let me explain you what is a fluorescence. We talk so much about the fluorescence microscopy, so let us first understand what is a fluorescence. So fluorescence is a two-stage chemical process. The first stage is the absorption of shorter wavelength of light, which is known as excitation, by a chemical fluorophore, followed by the release of some of the absorbed energy as a longer wavelength light, which is known as emission. So I'm sure you must have studied this phenomenon in your physics class, that when light interacts with matter, the electrons from the ground state, they gets excited and they move to the higher orbit, and then they fall back to the lower orbit by releasing the light of a longer wavelength, which is known as fluorescence. And this process is so instantaneous. You can see the time scale at 10 to the power minus 10 to 10 to the power minus 7 seconds it takes for this phenomenon. So it is almost instantaneous. The moment you emit the excitation light, instantaneously the fluorescence is produced. Now, how this physical phenomenon can be integrated in biological systems? So in 2008, these three scientists, Osamu Shimomura, Martin Shelfie, and Roger Weil, they were awarded the Nobel Prize for this Nobel Prize in Chemistry for their discovery of green fluorescent protein from the jellyfish Aquaria victoria. So you must have seen this kind of jellyfish. Under the seawater, it absorbs the blue color light of 488 nanometer, and it emits the 510 nanometer, the longer wavelength light, and that's why we can see them as a green colored creatures. Okay, so why only this fish gives this kind of a fluorescence? Because we also have several proteins in our body, but we do not fluoresce. Why only this fish gives the fluorescence? Because of the presence of a green fluorescent protein in this fish. And why this particular protein gives the fluorescence? So because of some specific arrangement of the 3 amino acids which produce this kind of a fluorescence phenomenon. So that we will study in some of the next class. But because of the presence of green fluorescence protein, this jellyfish Aquaria victoria, it gives green fluorescence under the seawater. So upon discovery of this GFP from this fish, it revolutionized the field of fluorescence microscopy, because now we can integrate this physical phenomenon into the biological systems. Another important thing, that why fluorescence is important. Okay, so in this figure you can see, if I ask you to count the number of stars in this image, which one you will prefer? You will prefer the one which is at the most left, or the one which is at the right hand side? So the answer is, you will obviously prefer this image where the background is dark and your particles are giving light. Okay, so in fluorescence microscopy, the background remains dark and only your object gives the light. So because of the good signal to noise or the better contrast, it makes it easy to quantify those information and to see those structures. Another important advantage that is being offered by the fluorescence. Weak signal against dark background is easier to measure, and high signal to background, that means a better contrast, as we discussed. So here you can see, here is the image of a cell taken in the bright field mode, and here an image of a cell that is taken under the fluorescence mode, and then the intensity was measured. So this line represents the intensity measurement area, and you can see from the bright field image, if you measure the intensity, you cannot see the structures which you are seeing under the fluorescence mode. But in the fluorescence mode, you can actually visualize this kind of protein localization intracellularly. Okay, so fluorescence offers better contrast and it makes the quantification easy. Another advantage is those are being offered by the fluorescence. So in this image, you can see the nuclei are stained in blue color, the DNA is stained in blue color, the microtubules are proteins and they are stained in green color, and the Golgi apparatus are stained in red color. So using 3 different dyes, we can label 3 or 4 different things in the same cell. So it offers selective labeling, because such kind of selective fluorescent dyes are available, which can be used for labeling various proteins or the cell organelles. Then this is also known as multiplexing, because we are seeing multiple color at the same time. So this is known as the multiplexing. So fluorescence offers multiplexing. If you are using the bright field microscopy, there is no way of multiplexing. Okay, and also this multiplexing is also quantitative. So from this image, you can say that here there are more number of Golgi apparatus are there compared to this cell, whereas where there is no Golgi apparatus, and this cell shows less number of Golgi apparatus. So this information is also called quantitative. You can also quantitatively measure the fluorescence intensity of different proteins and the nuclei to understand the quantitative, to get some idea about the quantitative biology. Okay, so these are the advantages being offered by the fluorescence, and that's why fluorescence microscopy is preferred over bright field microscopy in life science research. So as we are continuously talking about light and fluorescence, so let us understand some fundamental properties of light. Okay, so light is nothing but the electromagnetic spectrum. And if you see any physics textbook, all these different wavelengths from 10 to the power minus 12 meters to 10 to the power 3 meters, all these wavelengths are considered as a light. But generally for the fluorescence microscopy applications, we only use this small fraction or the small spectrum of visible light. So, as you know, the visible light is made up of these seven colors: violet, indigo, blue, green, yellow, orange, and red. So, the visible spectrum of light is made up of these seven colors, and it ranges from 380 nanometer to 700 nanometer. And only our human eyes can see the visible spectrum of light. Our human eyes cannot see the X-rays or the gamma rays or even the ultraviolet rays or the uh, microwave rays. Okay, so for the fluorescence microscopy, we'll be only using this light visible spectrum of the light. Some more properties of light. So again, going back to your physics class, and you must have studied about quantum mechanics and double slit experiment. Okay, so light shows wave particle duality behavior. So light behaves like wave as well as particle. Why we say that the light behaves as a wave? So this double slit experiment you must have studied before, that when you uh, uh, project these electrons on these two slits, ideally you should get only two slits on the screen. Okay, but instead of two slits you are seeing this kind of a diffraction pattern. So diffraction pattern diffraction pattern represents the wave-like behavior of the uh, electrons. Okay, now electrons are actually particles. Okay, but it shows this kind of wave-like phenomenon of diffraction. Let me introduce one new term known as photon. So photon is a minute energy packet of electromagnetic radiation, and it represents the quanta of light. And photon obeys the laws of quantum mechanics, and it shows wave particle duality behavior. And photon is detected as a single particulate unit. However, the probability of detecting a photon is calculated by equations that describes waves. Okay, so photon also have this wave particle duality behavior. If you want to find the probability of finding the photons, you need the equations those describes waves. But as such, photon acts like a particle because it is absorbed like a particle. So photon shows wave-like phenomena such as diffraction interference, and photon can be absorbed or emitted as a whole by atomic nucleus or the electrons. So in summary, just want to convey the concept that light shows wave particle duality behavior. Now coming to the useful size range for light microscopy. So as you can see, our human eyes can see any structures which are at or above 100 micrometer. But we cannot see if something is smaller than 100 micrometer. Our eyes cannot resolve if any structure is smaller than 100 micrometer. So the light microscopes can see the structures at and above 100 nanometers. Okay, and the electron microscope can see the structures at and above 1 nanometer. So see what you can see using light microscope. Using light microscope or the fluorescence microscope, you can see mycoplasma, bacteria, red cells, epithelial cells, so any kind of cells or the tissues that you can see using light microscope. But what you cannot see viruses, proteins, amino acids, and atoms using light microscope. Okay, so for seeing these amino acids, proteins, and viruses, you need electron microscope. Now there are various technologies available for fluorescence microscopy, such as live cell imaging, fixed cell imaging, time-lapse imaging, 2D imaging, 3D, 4D, 5D, and 6D imaging, super resolution imaging, single molecule imaging, high content imaging. So it is very important for you to understand when and where to use the right technology. Okay, so even though you may have access to the highly sophisticated microscope, maybe a super resolution microscope, but that doesn't mean that for whatever the applications you will only use super resolution, because it is possible that for some of your applications you do not need to resolve that much. Okay, but instead some other information is more important. So let us discuss that how to decide the best imaging technology for your experiment. So coming to the factors affecting the selection of the microscopy method. So see, the first point by which you can select your microscopy method is the type of sample. First you have to see whether you want to image bacterial cell, yeast cell, human cell, embryo, organoids, entire organism, or you are doing some in vitro assays or in gel assays. Okay, because if you are working with bacteria or yeast cells, you obviously need higher resolution, higher magnification. But if you are working with human cells, embryos, or organoids, you may not need that much magnification. Another thing to consider is imaging dimension. Whether you want to image in just X and Y, or you need the three-dimensional information so that you will image in X, Y, and Z. You will capture the entire Z volume of the cell, or you will image in 4 dimension, which includes X, Y, Z, and time, or 5 dimension, in which X, Y, Z, time, and multiple colors. Okay, so let me show you one example. Let's say we just looked at this image. Okay, now which image is this? This is a 2D imaging, 3D imaging, 4D imaging, or 5D imaging. So here you can see the X and Y coordinates of the image are recorded. Also, this kind of image needs the Z stacking because the entire cell volume is captured. Okay, it might be possible that in the three-dimensional structure of a cell, some mitochondria are on the top side of the cell, some mitochondria are on the bottom side of the cell. So if you just take the middle section of the image or of the cell, you may not see all the mitochondria. So what you have to do is you have to do multiple images throughout the cell, and you have to merge all those images to see all the mitochondria present in the cell. So this kind of image requires Z stacking also. So that is the third dimension. Now the fourth dimension is you are seeing the multiple colors. So that is the fourth dimension. But this is not a time-lapse movie. So the fifth dimension is absent. So this is known as the 4D image, which comprises of X, Y, Z, and multicolor. But let's say if this is a time-lapse movie, then it's a 5D image because it also has multiple time points. Okay, so this is how the dimensionality of the image can be decided. So you have to determine what you need, whether time information is important, that how the cellular processes are happening. Let's say you want to image the entire process of cell division, that how the cell is proceeding from prophase, metaphase, anaphase, and telophase. So in that case, you need to image the entire 3D volume of the cell along with multiple colors and at different time points. So that will lead to 5D image. Okay. Also in 6D image, you will also multiple positions on the same slide. So the multi-position will add to the sixth dimension. Okay. So come back to this slide, and the third factor that you should decide is how much magnification is required. So as I mentioned, if you are working with bacteria or the yeast cell, you need higher magnification. You will generally work with 60x or 100x objective lens. But as you are working with human cells or the embryo or the organoids, you maybe 20x and 40x objectives may serve the purpose. Okay. Again, you have to decide what resolution you need. Microscopes are available with 20 nanometer resolution to 300 nanometer resolution. But what resolution is required for your imaging application? So let's say, suppose this is an image of various diatoms. Now, if my research question is just to count the number of diatoms or just to see the size and shape of the diatoms, I do not need a better resolution. Even with 200 nanometer or 300 nanometer, I can see the size and shape and I can count the number of diatoms in one field. Okay, but let's say if I really want to structures of these diatoms, then I need a better resolution like 100 nanometer or 20 nanometer. Okay, to resolve the structures in a better way. Also, you should consider the fluorescent labeling and its bleaching rate. Like fluorescent proteins or the organic dyes are used for fluorescence imaging, and they do have bleaching properties. Okay, they do bleach. So you have to figure out what is the bleaching rate of your fluorescent dyes or the fluorescent proteins. If they are bleaching faster, you should go for some mild imaging technologies such as uh, at multiphoton imaging or the light sheet imaging. And if the label is your fluorescent label is quite stable, you can go for the epifluorescence illumination. Those things we are going to discuss in one of the next class in detail. Also, it depends whether your samples are live or fixed. For live cell imaging, you require to maintain the temperature, carbon dioxide, humidity, etc. Whereas for imaging of the fixed cells, you do not need to maintain any environmental conditions. And the last but the most important thing is you need to determine whether you need the fast imaging at every 10 milliseconds or you need slow imaging. Let's say every 10 minutes per hour or every 5 minutes per hour. So suppose you want to image the entire process of cell division, then that process takes, let's say, 1 hour. Okay, so it is okay if you image at every 5 minutes up to 1 hour to see how the cells are actually segregating the chromosomes or how the cells are dividing. But let's say if you want to study how the protein binds with DNA, and that protein binding to DNA is quite a fast process, so you may need to image those phenomenon as at every 10 millisecond interval. Okay, so imaging speed is also one of the very important factor in determining your microscopy technique. So with this, let us end this class today, and here are some online resources for further reading, and our next class will be on introduction to optics. Thank you.